let's talk about evidences of evolution. Coming to the evidences of evolution. So, usually there are many sources for evidences of evolution. First thing is paleontology. And second thing is comparative anatomy. Comparative biochemistry. And we have the comparative embryology. So you can find out very easily like comparative anatomy, adaptive radiation with A. Artificial selection. So we have the comparative biochemistry, and comparative embryology. So these are the things will comes under the evidences of evolution. Paleontology, comparative anatomy, adaptive radiation, artificial selection, comparative biochemistry, and comparative embryology. The very first thing we are going to see is paleontology, the study of fossils. We call it as paleontology. So coming to the paleontology, if you are studying about the fossils of plants, we call it as paleobotany. If you are studying about the fossils of animals, we call it as paleozoology. So let's talk about the paleontology. So coming to the paleontology, so the fossils are defined as remains of hard parts of life forms. Hard parts of life forms found in rocks. Fossils can be completely remain. So for example, completely you can see the entire fossil, such as woolly mammoth frozen in ice, insect encased in amber. If you see the movie Jurassic Park, you can see the mosquito is been trapped or encased in amber. Woolly mammoth in ice. Insects encased in amber. Hard parts, bone, teeth, petrified remains as cast and molds. So impressions, leaf impressions, feather impression, footprints, coprolites means fossil excreta of animals, coprolites. So these are all the fossils we call it as. So and fossil laid in sedimentary deposits means so sedimentation means the layering of the soil. So laid down in sedimentary deposits mainly. So over millions of years ago on history of Earth. Over millions of years in Earth history. If the sedimentary rocks was not disturbed due to earthquakes, the older strata will be deepest and usually contain simple and most ancient organisms. The younger ones will be on the topmost and usually contain complex and more recent organisms. This interference from fossils are, so you can see if sediments are undisturbed. Older ones are on the top. New ones on the superficial layers. Right? So let's see what and all the things we can able to see. So some of the life forms were restricted to certain periods of geological time. So you can able to see those organisms on the sediments of the particular era. For example, dinosaurs. Dinosaurs. So during Mesozoic era. Only in Mesozoic era you can able to find the dinosaurs. So we have the Paleozoic era, Mesozoic era and Cenozoic era. In that the Mesozoic era only you can able to find the dinosaurs. And geological age when the fossil arose. So geological age when the fossils arose, flourish and disappear. One of the most useful methods for determining the age of fossil is radiometric dating by using uranium, potassium and carbon, means half-life, based on the discovered fossils. So mainly we call that as carbon dating. We can also do with uranium and potassium as well. Geologists use radiometric dating to estimate the age of rocks, to interfere the age of fossils contained with those rocks. So radioactive atoms decay at a predictable rate by measuring the quantity of unstable atoms left on the rock, comparing it with quantity of stable daughter atoms in the rock. Scientists can estimate the amount of time that has passed since the rock are formed. So different life forms arose at different times. Life forms different times. So decreeing of difference between ancestor and present day organisms. Ancestors of modern day horses are vary from their descendants and that depict progressive development like size of the organism. If you see, for example, ancestor modern day elephants are also different from present day, which depicts the retrogressive evolution. Some modern day organisms will appear similar to their ancestors. Some fossils indicate absolutely no apparent change between ancestor and present day. We call that as living fossil. One of the example what we know is Limulus is said to be the living fossil. Some fossils acts as a connecting link between two taxonomical groups. So for example, fossil remains extinct, that is Archaeopteryx. Archaeopteryx exhibit both reptilian and avian character. So it has the both. So suggesting evolutionary origin of birds from the reptiles. So this Archaeopteryx is the connecting link between reptiles and birds. So they found that like birds, they got originated from the reptiles. So let's talk about the other thing coming to So let's talk about the next thing, from evidences from comparative anatomy and morphology. So a comparative anatomical studies of any particular organ or particular organ system is diverse members of given phyla reveal the basic similarity in them. For example, it varied to some extent from one class to another class. So usually from each phylum to another phylum there will be some similarities, skeletal, muscular. So if you see the systems, skeletal, circulatory, excretory, system of vertebrates, provides linear illusions. So coming to the two types of organs, first thing is homologous. Homologous organs. So you will be having same type of origin, but they are doing different functions. So the structure, everything will be similar, but they will be performing different functions. And we have one more category, which is analogous organs. So they are organs belonging to two or more different organisms. They are similar in origin. So you will be having similar origin. Here you will be having different origin. And internal structure, similar origin and similar internal structure also. Here also different origin, different internal structure. Homologous organs arise due to, we call this as divergent evolution. So why? Because it will perform different functions. It will perform same function. Like for example, so divergent evolution, in ancestry was common. So it will be having a common ancestry which is homologous, but you will be having different functions. So this we call it as divergent evolution. Homologous organs or divergent. When it comes to analogous, you will be having different origins, but all together it will perform same function. Okay, right? All together it will perform the same function, which we call it as analogous organs. So let's talk about the examples of analogous, homologous and analogous. So forelimbs of whale, bats, cats and humans. Their internal structure and origin will be same. But the forelimb of bone pattern of all the tetrapod vertebra have same anatomical structure, having humerus, radius, ulna, carpals, metacarpals and phalanges. But the basic structure has been modified in each species in a different way for different needs, adapted to the method of locomotion for a given environment. You can see for whale, bats, cats and humans. So different needs. So but due to these animal adaptation to the new environmental condition, they develop into different way in relation to the specific function. So forelimbs of man will have fine motor skills. Bat is for flying and cat is for running. Whale is for swimming. So if you take whale, forelimbs of whale is for swimming. Bat is for flying. Cat is for running. And humans is for fine motor skills. And in also plants like homology exhibited in plants such as thorns of bougainvillea and tendril of cucurbita are homologous. They both came from axillary bud. They both came from axillary buds, but they do perform different functions. So vertebrate of brains and heart is also homology. Vertebrate brain and hearts. And next, let's talk about analogous. Analogous will be having anatomically they are dissimilar, but functionally and externally they are similar. So, but they have erased different ancestry and indicate to finally as convergent one point. So, this we call it as analogous or convergent evolution. For example, wings of birds, wings of butterfly, they both will meet at one point, flying. So look, have same function, same like structure, but they have different anatomy. The eye of octopus and eye of mammal. They both have different origin, but they both are visual sense organs. Flippers of penguin and dolphin. Flippers of dolphin. So this both also, so penguin and flippers of dolphin, they both help for swimming. So fins of shark and porcupine. And fins of porcupine. They both have also protection. And we have the sweet potato and potato. Starch storage. Like jointed legs of insects and land vertebrates. So these are the things will combinedly you can remember. So and let's talk about vestigial organs. So coming to the vestigial organs, they are functionless. Remnant of functional organs. Functional organs in our ancestors. So like you can see in related species are, for example, Plica semilunaris, vermiform appendix, coccyx, rudimentary body hair, wisdom teeth, so are some of the vestigial organs are seen in man. Okay, wisdom teeth, coccyx, and we have the appendix, vermiform appendix, plica semilunaris, and rudimentary body hair. The presence of vestigial organs indicates the evolutionary history, relationship between organism and the ancestors, how functionality is lost. Reappearance of vestigial organs is called as avastism. So all this reappearance we call it as avastism. So next let's talk about evidence from embryology. So coming to the evidence from embryology, embryology is none other than the study of embryo and development. Evidences from embryology is based on comparative developmental studies of embryo. Okay, so of various organisms. For example, the developmental stages from zygote to gastrula in all the metazoans are similar. Carl von Baer, so the scientist name is Carl von Baer. So in 1928 studied young embryos in many vertebrates and noticed they are all similar, but they are difficult to distinguish from one another. He also said that during early embryo development, general characters such as appear before the special characters. More general or the less general, finally the special characters will appear. Like biogenetical law, we call it as recapitulation theory. So we have a law which is biogenetic law or recapitulation theory was proposed by Ernst Haeckel by studying the vertebrate embryos. So Ernst Haeckel, we know the Protesta, from the development of the kingdom Protesta. Ernst Haeckel has formulated the theory called recapitulation theory or biogenetic law. So the law states that by studying the vertebrate embryos, the law states that ontogeny, early embryo development, recapitulates the phylogeny, the evolution of race. According to Haeckel, early embryos in vertebrates recapitulates or recall the evolutionary of their race. And next, evidences from comparative biochemistry. So occurrence of identical macromolecules among the group of organisms indicates their evolution from common ancestor. So we have identical macromolecules. For example, you can see the hemoglobin HPG for humans as well as for chimpanzees have identical sequence of amino acid like alpha and beta chains, and differ from hemoglobin of gorilla by one amino acid each in alpha and beta, and by 3 amino acids in alpha and beta chains of hemoglobin of gibbons. So the similarity is extended by the genes to the genes too. This biochemical similarities indicates the common ancestry to the same way as anatomical homology. So this we call it as, so similar to anatomical homology. And next thing is evidences of from artificial selection. So evidences from artificial selection. So what are these evidences from artificial selection is, so usually human civilization is linked to domestication of animals, cultivation of crop plants and ancestral wild varieties. Though continued selective breeding, man has produced a number of varieties of domestic animals such as cattle, pig, fowls, dogs, cereal crops, etc. If man can produce a number of varieties within hundreds of years, so the nature can exert through selective breeding, but the nature can do over the same over millions of years. Human can do for hundreds of years. So for the nature, it takes millions of years. Nature can exert selection pressure over millions of years. Countless number of generations can produce new varieties and species. This above is an example of artificial selection. There is also an deliberate anthropogenic evolution, for example, non-deliberate or natural cum anthropogenic evolution. So the very best example for this is industrial melanization. Industrial melanism. So what is this industrial melanism is? So Dr. H.P.D. Catwell carried out the observation of experiments on peppered moths. Its name is Biston betularia, peppered moths. So the moths that exhibited the phenotypic polymorphism. It has two distinct morphological. One is darkly shaded. So it has two varieties. One is darkly shaded. Under 1948, light moths are predominant in England. So light shaded. Under 1848, light color moths are predominant in England. They are camouflaged against the light background. So because the tree trunk is covered by lichens, the melanic moths can be easily detected by diurnal birds and consumed. So darkly or easily detected or melanized moths are easily detected by diurnal insects. So melanic moths can be easily detected under the white background of the diurnal birds and consumed. However, after industrial revolution, pollution gases lowered the lichen's growth and got deposited on the bark trees. Hence, the melanic moths has an advantage and their number got increased. This assumption was tested by Kettlewell in 1950. He released the known number of marked dark and light and dark varieties into two areas: polluted area of Birmingham and unpolluted area of Dorset. Later he recaptured the moths and found that in polluted area nearly 34% of dark variety and 16% only light variety as could be recovered. In unpolluted areas, like recovery was 6% and 12.5%. So 6% of dark variety is there, but light variety is 12.5%. This showed that man-made event like pollution caused this evolution. So for an example, other examples, the anthropogenic evolution also included DDT resistant mosquito. And warfarin resistant rats. Antibiotic resistant bacteria. Drug resistant HIV. Drug resistant plasmodium. So finally we will see about adaptive radiation. So what is adaptive radiation? So the evolution of descendant species from an ancestor. So we have a descendant species, but from an ancestral species starting in a geographical point and laterally radiated into new ecological niches. We call that as adaptive radiation. For example, when the descendants become geographically isolated by barriers such as sea, river, mountain from the parental population, they may exploit to different habitat of new area and become evolved into number of separate species. Darwin observed the adaptive radiation in a small blackbirds, later called Darwin finches. So adaptive radiation we have two examples. Darwin finches. And the next thing is Australian marsupials. So first thing is Darwin finches. So Darwin finches, he studied in Galapagos Island. He could observe the large variety of finches, suggested that all these varieties evolved from common seed-eating group finch ancestor. So Darwin finches evolved from seed-eating ancestor. So the ancestral finch evolved into insect eating. So from the seed eating they got insect eating. Camericus species and vegetarian finches. So insect eating, vegetarian finches. Marsupials of Australia is also an example of adaptive radiation, right? So coming to the marsupial, we closely related which when too closely related but individual species undergo adaptive radiation among gives rise to similar species. The term we call it as parallel evolution. So, adaptive radiation will exhibit independently by the marsupials in England and eutherian mammals in other parts of the world. So, eutherian mammals. Surprisingly, the pouched mammals of Australia closely related with the placental mammals in Australia or to the rest of the world. They exhibit parallel evolution. Parallel evolution occurs in different isolated geographical area due to similar selective pressure under identical environment. Each pair of similar species represent an example of convergent evolution. So let's talk about the ancestor. So let's talk about Australian marsupials. And we have the eutherian mammals. Tasmanian wolf. And here we have the wolf. Tiger cat. Bobcat. Flying phalanger. Flying squirrel. Spotted cuscus. Lemur. Marsupial mouse. Marsupial mouse, and here it is mouse. Marsupial mole. Marsupial mole, here it is mole. Banded anteater. And here also anteater. So you have to remember spotted cuscus and lemur. So these are the similarities between, these are the organisms which look same. This will comes under convergent evolution, because whatever the ancestor that got separated by ocean, sea or land, under same similar conditions what they have exhibited, they had similar type of adaptive radiation, adapted to the same type of thing. So this is the parallel evolution, or you can also call it as convergent evolution.